Hello, everybody. I am very pleased to welcome Wakuna M. Galega from sunny Stanford University, where she's a PhD candidate in the Environmental and Engineering Department. She's working with Professor Craig Criddle, and her research focuses on the microbial degradation of methane in mixtures such as biogas and natural gas for the production of biodegradable polymers called polyhydroxyalkanoates, or PHAs. Wakuna is interested in understanding the impact of these methane-rich mixtures and what they have on the microbial communities, the dynamics between the microbial interactions under certain complex conditions, while optimizing the polymer production process and the bacterial growth rates. So I will stop my share, and Wakuna, you can put up your slides for us, and we will be ready to go.
Can you hear me?
Yes, wonderful.
Great. Hi, everyone. My name is Wakuna. Holly, thank you for that introduction. And today I'm going to be presenting my work that's going to tell you a little bit of a story about how methane-eating bacteria can sequester carbon dioxide while making biopolymers. And first, I want you to take a closer look at this map of the US. And this particular image I pulled out of the 4th National Climate Assessment. And what really fascinated me in this figure was seeing how the impact of climate change seemed to have overwhelmed the US in just under a decade, from wildfires to droughts and longer seasons, hurricanes, floods. It just seems like we were hitting all the indicators of climate change here. In fact, the recent emissions gap report from last year even highlighted that the global CO2 emissions had started rising again after a brief period of stagnation. So for me, what this says is, even with all the policies in place to mitigate greenhouse gases, the key message this report, I believe, was trying to relay was more of a call for action to researchers and scientists like myself, asking us to increase our efforts in battling this greenhouse gas problem. So the question becomes, how do we scientists make carbon dioxide a resource and not a problem? My research specifically looks at carbon dioxide in the context of methane mixtures and how these mixtures can be used to make valuable products. So one of these mixtures is biogas. Biogas, as many of you might know, is a byproduct of fermentation and is made up primarily of methane and CO2. And this biogas could originate from a wide variety of sources, as I have highlighted a few here: landfill, sewage, and the rest. And based on their waste content, the methane and CO2 compositions could vary over time. Natural gas as well is also another one of these mixtures that I am interested in, that contains CO2, but in a very small quantity. But for this specific study, however, I'll be focusing more on simulated biogas compositions. So going into the goals of my study, I wanted to understand the impacts of supplementing carbon dioxide in the feed of methane-eating bacteria and how this affected its growth and in turn its biopolymer production. So for this talk, I would start by giving you a brief introduction about these methane-eating bacteria and the polymers that they produce. Then I'll go further to talk about the objective of the study while pinpointing some key findings from this study, and I'll end with the implications of my work. But just to give you a big picture overview to start with. So methane-eating bacteria are known. They are also called methanotrophs, as you can see here, and they are known for their ability to grow on methane and accumulate biopolymer. This is almost state of the art for them right now. And these polymer are naturally produced, which means they are biodegradable, and they serve as excellent replacement for synthetic polymers, given that their mechanical properties are quite good as well. But when extracted from the bacteria, since they accumulate within the bacteria, they can be used to make a wide variety of plastic products. My current study, however, would look specifically at how I can use these biogas mixtures to make biopolymers, and how understanding really the effects of having carbon dioxide in the mix of the raw material. But what makes my process really interesting is that it's a closed loop cycle. So the biogas, when used to produce the bioplastics, at the end of life of the bioplastics, because they are biodegradable, they can be digested anaerobically to produce more biogas to continue the cycle. The cycle can also be shortened through a recycling loop, where the waste plastic can go through a hydrolysis or pyrolysis process, broken down into some polymer precursors, which are then fed back to the bacteria. These bacteria have the capabilities of taking up those precursors to make biopolymer of the same quality. And in this case, we don't have to worry about any downcycling of the plastic. So what are these methanotrophs or methane-eating bacteria, and how is this process of biopolymer production possible with them? So methanotrophs are organisms that are specialized in completely oxidizing methane to CO2, as you can see, through a series of reactions here, which means that they use methane as a sole source of carbon and a sole source of energy. And there are three main types of methanotrophs, also highlighted in the boxes. Type 1s, type 2s, and type 3s, and they assimilate carbon in that process of methane oxidation through a number of branch points, as I have highlighted in these figures here. And of these three classes of methanotrophs, only the type 2s are known to date to accumulate biopolymers. The biopolymers themselves are called PHAs, and they are polyhydroxyalkanoates. That's how they pronounce. And these PHAs are a family of polymers made of carbon, hydrogen, and oxygen, and their chain length could vary based on the bacteria, the conditions, and different conditions, and different things would cause that chain length to vary. Their ability to degrade naturally and their excellent mechanical properties, as I mentioned earlier, is what makes them very attractive. But they are primarily produced by bacteria, and they accumulate intracellularly in the bacteria. So how does this happen? When the bacteria are fed everything that they need in terms of nutrients and elements for growth, they would not produce PHAs. But in order to trigger PHA accumulation, they need to be starved of some nutrients. And in most cases, the strategy used is nitrogen starvation. So in the case of methanotrophs, when methane is fed as the sole carbon source, PHB, also called polyhydroxybutyrate, which is one of these types of PHAs, is the polymer produced. The major limitation, however, with PHB is that it's very brittle in nature, which means the range of applications that it can be used for is very limited. However, to improve these properties of PHB, we can add co-substrates, some organic acids, that can make the polymer a little bit more flexible alongside the methane, to make the polymer a little bit more flexible. So, but because I show you these two, PHB and PHBV, it doesn't necessarily mean that they are the only two types of PHAs that are possible. There's a wide range of them, but my talk will be looking at these two, and you would understand why as I move forward. So to give you an idea of what these PHAs can be used for, the applications are quite diverse. So they can be used when extracted out of the cell. They can be used in a whole variety of plastic applications in the medical industry. In fact, one of the PHAs has been approved by the FDA to date for fabricating medical implants. They can also be used in the fuel industry. One discovery that we've also found out, and it's work that is going on in my lab, is that without being extracted from the cell, you can feed this PHB to aquaculture, shrimp, and that's going to boost their system, and it almost acts like a prebiotic to them. So these plastics are really very attractive. So what I show you on this next slide are the key metabolic pathways in this type 2 methanotrophs that make PHB. So for a long time, methanotrophs have been known as methane consumers alone. But recently we've discovered that they also have the ability to take up CO2 through those inlet points that I highlight in the red arrows. In fact, a recent study revealed that about 60% of the cell biomass came from CO2 alone. And so we became curious about how CO2 could impact PHA production. This was therefore the motivation of my study. And in this study, I look at the impact of CO2 using two type 2 strains to get a better understanding whether the impacts that I would see from one strain to the other would be universal. So now I go into the results of my study. One of the most outstanding results was the decline in the CO2 generation. As you can see here, I'm showing you graphs during the growth phase of when CO2 is fed at different increments of CO2 in the feedstock or ratios, and during PHB accumulation. And what we observed was as we increased the CO2 concentration in the feed, we generated less and less CO2, whether we were just growing the cells or accumulating PHB. In fact, at some point we got to negative production, which indicated that the cells were actually consuming the CO2 that was being provided, which means CO2 at that concentration that was, or that methane to CO2 ratio was being sequestered. So what could be happening in this case? As I initially highlighted, when methane is oxidized, the final product is CO2. It is possible that the last gene called formate dehydrogenase that is responsible for producing CO2 is either being repressed or downregulated because of the external source of CO2 available. And that way, most of the carbon from the methane is being channeled into growing the cell or accumulating biomass. But one thing we know about CO2 fixation is it is a very energy expensive process. So it was important for us to look at the energetics of the process to properly understand the energy dynamic, which then brings me to this oxygen consumption chart here. Here, oxygen consumption is used as an indicator for energy demand. And what we notice is we see almost an increasing progression as the CO2 levels increase. It looks like the amount of oxygen that is consumed increases. So when methane is oxidized, 2 moles of oxygen is required to completely oxidize 1 mole of methane. 2 moles of oxygen required to oxidize 1 mole of methane. And 1 mole of methane alone goes towards just activating, as you can see in that reaction, that first step of methane oxidation, which makes the process initially from the get-go very energy demanding. So with CO2 addition, we observe even an increasing energy demand with increasing CO2 levels, which therefore confirmed our hypothesis about the high energy requirements for CO2 fixation. But then the next question for us was whether CO2 was actually being incorporated into the biopolymer itself. And the straight up answer is yes, it did end up in the polymer. So what you see here is an NMR spectra showing the different positions of the carbon in the biopolymer. And for this analysis, we used 13C isotope labeled CO2 and methane. So the peak heights shown here are the intensities of the carbon under different incubation conditions. And from this spectra, what we noticed here is what I'm showing you is what the PHB polymer looks like, is that the CO2, 13C CO2, ended up in the C1 and the C3 positions of the polymer, while 13C methane, on the other hand, ended up in the C2 and the C4 positions of the polymer. So in here, it is possible, we believe, that it could be a thing of convenience for the cell, because if you look at those bonds, they look, especially with the CO2 ones, it looks very similar to CO2, and where the methane ended up, it looks very similar to methane. It could also be potentially very thermodynamically for the cell to pick that kind of positioning. But since very little thought had been put into this prior to my finding, we decided to track the carbon through the metabolic pathway itself to confirm our observation. And we did this little test. So I'd like you to pay attention to the green and the orange dots while I walk you through this pathway really quickly. So we start with methane at the very top, and the methane carbon, the orange dot, gets into the serine pathway as formate and goes through a series of transformations until it gets to PEP, which is phosphoenolpyruvate. And here a CO2 inlet point is introduced. At this CO2 inlet point, CO2 then gets in the green dot to form oxaloacetate. And oxaloacetate again goes through a number of transformation and ends up in, you see the carbon from the methane and the CO2 ending up in acetyl-CoA, which is then the beginning point for PHP accumulation. Acetyl-CoA now, which contains the carbon from methane and CO2, continues all the way to then making PHP, and there you have it, the methane and CO2 positioning matched our NMR results. This was quite interesting. We were also interested in understanding the impact of the CO2 on PHPV, which I mentioned previously was a more flexible polymer. So to just give you a quick recap here of the process, the PHA polymer is accumulated within the bacteria. When methane alone is fed, PHB is produced. Now to improve the properties, we add a co-substrate, and in this case, we add valeric acid to make PHBV. And this PHBV has an HB fraction from the methane and an HV fraction from the valeric acid. So what did we observe from here? Using the same methane to CO2 ratios as before and just adding the same amount of valerate in every different scenario, what we observed was that HV fraction of the polymer increased progressively as the CO2 concentration increased over time. Again, we had to ask ourselves what was happening, and to get a better understanding of what was going on, we had to look at the pathways that assimilated, that led to this process to come up with some kind of hypothesis. So I show you another pathway here, and I'm going to walk you through it. Here, when valerate is added, it is transformed through a process called fatty acid beta oxidation to produce two major precursors, acetyl-CoA and propenyl-CoA, which together combine to form a 5-carbon precursor, which makes the HV fraction of PHBV. But also the EMC and the PHA cycle of the methanotrophs itself has the propenyl-CoA metabolite, which is the same one as the one that is produced from valerate fatty acid beta oxidation. You would also notice that upstream of the propenyl-CoA, that is the metabolite of the methanotrophs, there are a number of CO2 inlet points in red. And the hypothesis that we drew out of this was that it's possible that this intracellular propenyl-CoA concentration was building up with increasing CO2 and then contributed to that PHBV. However, at this point, this is still a hypothesis, and we hope to test this in the future. There is also a downside to CO2 fixation. We observed that even though that HV fraction in all the different scenarios was increasing, the total amount of the PHBV that accumulated within the cell peaked at a methane to CO2 ratio of 2:1 and then dropped thereafter. And we believe this happened because of the energy demanding nature of the elevated CO2 levels, costing the overall PHBV accumulation. So that brings me almost to the end of my talk, and I'm going to summarize the findings here, I demonstrated decreasing CO2 generation, which led to CO2 sequestration by the methanotrophs. And then this in turn also demonstrated an increasing energy demand because of elevated CO2 levels. CO2 was indeed incorporated into the PHB in a unique positioning, and it affected the HV fraction of the PHBV polymer. One thing that I want to highlight about PHBV is the greater the fraction of the HV, the more flexible the polymer is. So you really want a PHBV polymer that has a high PHBV fraction. So moving on into some practical implications of using a methane CO2 mix as feedstock. Using a methane CO2 mix as feedstock, sorry, I'm a bit distracted here. Well, I say on Earth because one thing I've shown is that by using methane CO2 mixture to make polymers, we mitigate a very crucial greenhouse gas and sequester it into a product. But because methane CO2 is generated from waste sources, it is a very low cost feedstock. In fact, it's even much cheaper than having to use methane alone, because with methane alone, you would have to kind of clean up the gases of any impurities to use methane alone. But finally, with changing environmental conditions, you could almost imagine a scenario where methane and CO2 ratios are changing over time and the methanotrophs are able to adapt to such changing conditions. Because this project is sponsored by a NASA grant, they wanted to be able to make products for space explorations that would sustain astronauts while in space for extended periods of time. And Mars, we know, is almost 90% CO2. So by using a resource that is already available, we cut the cost of the feedstock for biopolymer production, and they won't have to travel with their own feedstock while going up there. But then because this is a recyclable process, the astronauts, after making their own biopolymer, could just anaerobically digest the waste plastics that they don't need at the end of the day with their waste to make more byproducts, to make more raw materials, to make more polymer. So where are we going next with this? I'm sorry, I want to short this. The first thing that we want to do in this project is a few extensive genetic studies. One, to confirm some of the hypotheses about the downregulation of the genes I mentioned and the building up of some of the genes intracellularly, just to give us a better metabolic understanding of what was going on within the cell. But also to understand the energy demands of the cell and think of ways that we can supplement the energy that is needed to cut down the cost of the overall process. But also more importantly, methanotrophs are very slow growing. And in order to make large-scale processes, we need to think of more creative ways, whether reactor design, but how can we optimize this process so that it can be more attractive in the commercial sphere. But long-term, we want to look at the implications of running this system for longer periods of time. This was done in a laboratory setting, so we want to be able to simulate processes where it is almost like an ongoing PHB producing process, but also understanding the impact of using actual biogas from different sources. One, we can't predict what the methane to CO2 ratios would be from all the different sources, but also actual biogas, because of the downstream processes, has some waste in it. So how does the methanotroph handle all of that waste would be something that we'll be interested in. That brings me to the end of the talk. I'd just like to acknowledge my lab, the Credo Lab, and my collaborators lab, the Weymouth Lab, and the grant that sponsors this project, the NASA grant under the CUBES. project. So thank you very much. I am ready for any questions.
Oh, wonderful, Wakuna. Thank you. I'm so glad you were able to explain it at a little more layman's level because I actually was able to understand some of it.
Yeah.
And I have some questions. The first one says, very interesting. How long does it take to biodegrade the flexible polymer you spoke about?
Okay, so I have some additional slides. I wish I did, I don't have comparable to regular synthetic plastics, but synthetic plastics we know persist, so they stay for a long time. But this figure here just gives you an idea of some rates of degradation in different environments and different kinds of plastic. So this was categorized based on if you were just a plastic bags, like the bags you were using Walmart, or a straw or container or bottle, that kind of thing. And looking at the practical scales, it could range from months to years, and the max I could see there was for cutlery was for 5 years in a marine environment. Anaerobic digesters would definitely take anything from days to months based on the process. So those are some time scales that I got here.
Okay, thank you. Have any LCAs been carried out on PHA from methane?
Yes, actually. An LCA out of our lab a few years ago was carried out, and they really looked at the process from, they call it cradle to gate, because they looked at from raw material using biogas to the intracellular polymer itself. They didn't look at the extraction process, because the extraction process is pretty similar across different methods, and they were comparing biogas in this case to corn, and they found that using biogas methane when compared to corn was more preferable. But the major challenges was really, again, like I mentioned, recovering it, to extracting it out of the polymer, because it out of the bacteria, because it requires a lot of solvents, and the environmental concerns of disposing of large amounts of solvents, but also the energy demands, like I mentioned, in activating the polymer and all other downstream processes. But one recommendation out of that LCA was using a renewable energy source like biogas could almost meet some of those energy demands, and even reduce that CO2 equivalence number to about -6.
Okay, thank you. And Wakuna, how far away are we from commercialization?
We are almost there. There's still a lot of problems, like I previously mentioned, but we are almost there. So a few startups pretty much have started looking at this PHA from methane. So Mango Material is a startup out of our lab about, I want to say 6 years ago, and they're in San Francisco, and they started using anaerobic digester biogas to make PHP, and they're targeting cloth fiber. But again, with all the challenges and all of that, they have still maybe not marketed yet, but they have started the process. And there's a new company also called New Light Technology, which also claims to use a method of biogas and biocatalyst. And I think the biocatalyst in this case is a microbial biocatalyst. And they've recently just landed a deal with IKEA to produce biopolymers.
Thank you. Someone says, has there been any comparison of PHA and PHBs to methylcellulose?
To methylcellulose, I am not sure exactly if there's been any comparison with methylcellulose.
Okay.
Will the cellulose be a feedstock or product?
This was the only question I got.
Okay. All right.
Another question is, if this was to be turned into a commercial process, can you describe what you would foresee in terms of location, where the methane CO2 mixture would come from, and what are the major products that the facility would produce?
Right. So I think I'll start backwards and move forward. I think the major product would be PHB, just proof of concept to make sure that it works before then moving to better polymers. Based off of Mango Materials method or what they have employed over the years, they plugged themselves into an anaerobic digester plant. And that plant is now producing the biogas that they need for their process. I think it is going to be cost effective to associate yourself with whatever source of biogas you're producing, because one issue with biogas is the collection and the transportation to wherever you are. So to kind of cut some of that cost is building your plant around that. And then maintaining your consortium of bacteria to make your biopolymer, and after you can start thinking of ways to either add valeric acid. Valeric acid would be expensive, would come at a cost, but then thinking of other substrates that can be, or other organic acids that can be added to the feedstock that will be cheaper to make new different more flexible polymer. I think that would be the best first place to start.
Okay, and what are the first products that would be made from PHB?
Or the first products that will be made out of PHB? I think it could be anything starting from Tupperwares, anything containers, like anything that you can heat press and make a container out of, or cutlery even. I think those would be like the first things to start with.
Okay.
Yeah.
And does your process require biogas purification for H2S removal?
That's what I was mentioning earlier. So right now I'm using synthetic biogas. So I am mixing the methane and the CO2 mixtures in the lab because I want to have a more controlled process. But in the future, and I think Mango Material, I don't think they're working with pure cultures. In the future, if we wanted to work with pure culture strains, we would have to then test it with actual biogas to be able to see how the methanotrophs can handle some impurities like H2S. The goal is not to have to scrub the gases of any of those H2Ss if the methanotrophs can handle it, except otherwise.
Mm-hmm. Okay, thank you. Do you have any thoughts on how to solve the energy problem that you mentioned in the whole process?
Yes. So methanotrophs or a lot of bacteria are known to have hydrogenases, and we believe methanotrophs do. We've not been able to see them actually be activated because working with gaseous substrates is very hard because they don't dissolve very well. So we anticipate that we can try to engineer the organism in such a way that its conditions are favorable to take up something like hydrogen, which can then offset some of the energy cost, and then the methane carbon would be used solely for carbon assimilation. But that's the goal.
Okay.
Yeah.
Thank you. Someone says, does the polymer accumulate within the cells and then they would need to be lysed to collect the material?
That's correct. Yeah.
Okay. And the same person is wondering whether it's possible to get a copy of your slides, but I'm thinking they should just contact you directly
Yes.
to get that.
Yeah, and I would love to, but I can't because I'm in the process of publishing and I don't really know how
Right,
this work.
right, of course.
Yeah.
You have mentioned some of this, but I didn't know if there are any other major challenges you wanted to mention with your producing the methanotroph process, the PHA.
The main one thing with the methanotroph, one of the major challenges is mass transfer. Because of getting gases to dissolve in liquid culture. There's a mass transfer barrier that we need to breach. And one way to do that, or at least we've been thinking, is proper reactor design using more fermentative processes, which is really what causes the methanotrophs not to grow. very well because of that mass transfer issue. But that is one of the biggest challenges we really have now outside of the energy demand with methanotrophs.
Okay, I have one last question here. If anyone else has more questions, please send them along. What about natural gas? Is there any progress on methanotrophs making PHA from natural gas?
Yes, absolutely. So I mentioned earlier, and I'm actually working on a process that would use methanotrophs, and I'm seeing that methanotrophs can use natural gas to make PHAs. Actually, they can actually use one of the strains of methanotrophs that I mentioned earlier is able to use ethane alone to make PHP. So that was exciting because we also anticipate a scenario where we can plug pipeline natural gas into our process and still make PHP without the fear of toxic metabolites building up and intoxicating the organisms.
Great. Now, one more personal question for you. How did you get interested in this methane-eating bacteria?
I don't know. I mean, when I started looking out for a PhD project, I was more interested in a project that was more practical, and that was just going to challenge me a little bit. And this was the most outstanding, I think, is how I got into it. I went through a number of routes, but there really was the theme of making a more sustainable product from recovered resource. So that's what attracted me to this.
Well, good for you, Wakuna. I'm so glad we have people like you that are doing this sort of thing instead of people like me. So if you will stop your share, I will talk about the next event we have next week, and I will be getting ready to close this webinar. Next month, on April 25th, we will have Rebi Feraldi, she's an MS candidate from Arizona State University, talk about LCA and biomimicry. So please feel free to tune into that. And Wakuna, I thank you again for a wonderful program. So good luck to you. So happy that you can be there to do this for everyone.
Thank you so much.